Good day, students. Today we are going to talk about the single membrane bound organelles. In the previous lectures, we have discussed about the general introduction of the cell, the nucleus, the cell membrane, cell wall, and the double membrane bound organelles like mitochondria and plastids. Now we are going to start about the single membrane bound organelles. As the name implies, these organelles are enclosed within a single membrane, and this in the example of single membrane bound organelles include endoplasmic reticulum, Golgi complexes, lysosome, vacuole, etc. In the single membrane bound organelles, first of all we are going to talk about the organelle that is commonly known as the endoplasmic reticulum or ER. First of all, who discovered endoplasmic reticulum? It was discovered by three scientists, that is Potter, Claude and Fulham. In the muscle cells, endoplasmic reticulum is also known as sarcoplasmic reticulum. So we can say in the striated muscles, the word sarco means muscles, and striated muscles are the type of muscle cells which contain alternate light and dark bands. Therefore, their appearance is striated, that is alternate light and dark bands. Where the endoplasmic, in which type of cells the endoplasmic reticulum is absent? So we can say that ER is present in all the eukaryotic cells except the red blood cells. Also, ER is absent in the prokaryotes because we all know that the prokaryotes lack the membrane bound organelles. RBC stands for the red blood cells or the erythrocytes. Now coming to the structure of endoplasmic reticulum. Structurally, ER is composed of three types of components. So we can say it is made up of three types of components. And these are known as cisternae. Cisternae are the long, thin, flattened sac-like structures. So we can see that cisternae are made up of thin flattened sac-like structures which are made up of the lamellae or the membrane, and these cisternae are studded with the ribosomes, means on the surface of cisternae, ribosomes are present. The second type of component is known as the vesicles. Vesicles are the oval or the round structures and just like the cisternae, the vesicles are also studded with the ribosomes, that is on the surface of vesicles also ribosomes are present. The third type of the component or the element is called as the tubules. Unlike cisternae, the tubules are also the cisternae, the tubules are the much wider branched structures which form network-like structure. So we can say tubules are branched net-like branched elements which form net-like structures. And in comparison to the cisternae, the tubules are much wider in the diameter, and there is one more difference between the cisternae and tubules, that they don't have ribosomes on their surface. So unlike cisternae and vesicles, they lack the ribosomes. Now, after knowing briefly about the components of the structure of the endoplasmic reticulum, now coming to the types of endoplasmic reticulum. On the basis of the type of the elements present and function, the endoplasmic reticulum is of two types, and they are known as the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Smooth ER is also known as SER. And the second type, rough endoplasmic reticulum, is also known as the RER. Now let us see some differences between these two types of the endoplasmic reticulums. The first difference lies in the terms of the type of element present or component present. In case of SER, it is made up of mainly the tubules. Whereas the RER is made up of either the cisternae or the vesicles or both. Regarding the presence or absence of the ribosomes, in case of SER, the ribosomes are absent. As I have explained earlier also, that the tubules of which the SER are made, they don't have ribosomes on their surface. So in case of SER, again the ribosomes are absent. In case of RER, since it is made up of cisternae and vesicles, so ribosomes are present. And due to the presence of ribosomes, the appearance of the endoplasmic reticulum becomes rough. Therefore, it is known as rough endoplasmic reticulum. And due to lack of ribosomes, the appearance of endoplasmic reticulum becomes smooth, therefore it is known as smooth endoplasmic reticulum. Now regarding their functions, the main function of SER is synthesis of polysaccharides, steroids, lipids. In case of RER, that is rough endoplasmic reticulum, its main function is synthesis of proteins. SER plays one more function, that is detoxification of the antigens, whether they are endogenous or exogenous. What are antigens? Antigens are the foreign molecules or the molecules which are of either protein or polysaccharide origin present on the surface of the foreign molecules. So SER helps in the detoxification of such antigens. Now coming to the functions of endoplasmic reticulum, we have come to know about some of the functions when we studied the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. So broadly we can say the main function of the endoplasmic reticulum is synthesis. So its primary function is synthesis of variety of biomolecules like proteins, which is done by rough endoplasmic reticulum. Besides proteins, it also helps in synthesis of lipids, steroids and polysaccharides. Polysaccharides are also commonly known as the carbohydrates. As I told you earlier also, that the synthesis of lipids, steroids and polysaccharides is carried out by the smooth endoplasmic reticulum. Second main function, or we can say one more important function of the endoplasmic reticulum, is the circulation of the different molecules in the cytoplasm, different parts of cytoplasms. Since endoplasmic reticulum forms a continuous channel running from the plasma membrane to the nuclear membrane, thus it helps in circulation of different types of molecules from one part of the cytoplasm to the other part of the cytoplasm. So it forms a continuous intracellular transport system. Also, the ER provides the mechanical support to the cell by acting as an accessory element to the cytoskeleton. Both the functions, that is transport system and the support system, mechanical support system, function of which is the function of endoplasmic reticulum, is because the endoplasmic reticulum forms a continuous channel-like system running from the plasma membrane to the nucleus membrane. Also, the endoplasmic reticulum helps in membrane biogenesis. Especially the vesicles are involved in the formation of nuclear membrane at the telophase stage of cell division. So we can say ER also helps in membrane biogenesis. Also, the endoplasmic reticulum helps in detoxification of the antigens. and this function is performed by the smooth endoplasmic reticulum. Now, after talking about the endoplasmic reticulum, the next important single membrane bound organelle is the Golgi complex. We'll discuss about it in the next lecture.